Hi, everyone, and welcome to today's webinar on food spoilage. We're excited to have you here with us. My name is Jessica, and I'm the Director of Marketing and Sales here at Amtec. I'll be moderating today's webinar. First off, we encourage everyone to participate by asking questions throughout the webinar. You can submit questions by clicking on Q&A at the bottom of your screen, and we will save some time at the end to go over the questions that came through. To start off, for those of you who don't know, Amtec is an accredited laboratory in the San Francisco Bay Area, specializing in food safety testing and special research projects such as shelf life, challenge, validation, and of course, spoilage investigation studies. Amtec also just opened a laboratory in Modesto, California to better serve the Central Valley. So if you or anyone you know is looking for superior service in the Central Valley, please do not hesitate to reach out. Today's webinar will be led by Dr. Florence Wu, founder and president of Amtec. She has extensive experience in shelf life testing and spoilage investigations for food and beverage products. Florence has over 30 years academic and analytical experience in mycology and microbiology, and over 18 years of managing an independent contract laboratory. So with that, let's get started. Florence?
Thank you, Jessica. And welcome, everyone. Thank you for spending this hour with us to talk about the microbial spoilage of foods. I really like this webinar to be an interactive one, but in the interest of time, I will share the things I learned over the years, and then we'll open up for questions. As you may already know, food loss is a huge problem. It was estimated that around 1/3 of the world's food was lost or wasted every year. You know, personally, this morning, I had to throw away a whole bag of bagels because they are moldy. USDA ERS estimated that in 2010, in the US alone, 31% of available food supply went uneaten. The estimated retail value was $162 billion. This food loss, including loss from microbial growth, pest, improper storage conditions, cooking loss, and natural shrinkage and waste. And today we focus on microbial storage. Microbial spoilage is caused by the growth and fungi. In this type of spoilage, microorganisms digest the food to support their growth. And during the process, they generate byproducts of their metabolism, such as bad odor or acid or toxin, that make the food unsuitable or even toxic for human consumption. Often, unless the food is sterile, or the conditions of the food make microbial growth impossible, the microorganism population in food constantly changes. Some food can naturally inhibit microbial growth. Some food are formulated or produced with controlling microbial growth in mind. Basically, if microbial load in the food increases, the food will get spoiled. If microbial load does not increase, then it's unlikely a microbial organism is the cause of spoilage. If not controlled, microbial growth becomes more evident and can be detected by changes in odor, taste, color, texture, etc. The threshold levels for detectable sensory changes varies. Some data say that when bacterial growth is over 10 to the power 7 CFU per gram, you may notice the off odor. Some yeast can cause detectable bulging, even when their number is relatively low. Consumers often use the terms such as moldy, sour, bloated, fermented, or simply it stinks to describe spoilage caused by microorganisms. The spoiled food can look really awful too. So I don't want to ruin your appetite for lunch, so we won't show the really disgusting pictures here. The webinar topic is about microbial spoilage of food, and there are three factors we'll consider: microorganism, food, and the suitable conditions that promote the spoilage. We'll first discuss the major groups of microorganisms and how we classify and naming them, and based on the conditions of their growth. As we mentioned earlier, for bacteria or fungi to spoil food, they must be able to grow in the food. The food provides nutrients necessary for growth, but other factors can inhibit or promote microbial growth, such as water activity, temperature, oxygen, acidity, and preservatives, etc. These factors are so important that we can group microorganisms based on their growth requirements in related to water activity, pH, oxygen, temperature, and other factors. For example, based on microorganisms' requirement for oxygen, we can group them or describe them as aerobic, that require oxygen to grow, anaerobic, that does not require oxygen to grow, and something between, like facultative anaerobic, or requires certain mixture of oxygen or to grow, which is microaerophilic. And then based on their requirement for temperature, we described microorganism as mesophilic, that's normally grow happily between 20 to 45 degrees, and psychotrophic that prefer refrigerated condition to grow, and they cannot grow when the temperature is above 20 degrees. And then thermophilic are those can grow when the temperature is above 43 degrees. But all this degree is not really hard limit, is their range, their preference. And then based on their requirement for water activity, we describe them as hydrophilic, which means they need a high water activity to grow, or xerophilic, meaning they may be able to grow in high water activity, but they can also tolerate low water activity grow. And halophilic that require, can grow in some solutions that have high salt content, and osmophilic bacteria or yeast mold that can grow in high sugar content food. And acidity can be expressed as you say, pH or acid level. So based on that, every species or organism of bacteria or fungi has their range of pH. Outside of their range, they either cannot grow or they die off. And then there are some bacterial fungi are acid tolerant, while others, there are some species can actually prefer the acidic condition. So the first group we would like to talk about is visible mold. If you search Google Images for food spoilage, most of the pictures that pop up are moldy food, right? That's because mold growth on food are so visible and disgusting. And wearable mold usually indicated the end of shelf life, because no one wants to eat moldy food. Of course, specialty cheese with inoculated mold is another story. Mold can grow on low water activity and low pH foods on which bacteria cannot grow. So that's an important point there. So they have a wider range of growth. Common mold species have widespread distribution. So it is pretty hard to trace where particular mold contamination come from. Controlling water or moisture during the storage is the key to prevent the mold growth. I cannot emphasize that, enough. It's always the case with mold growth, and proper proper harvest and post-harvest protection are also very important. For example, as you can see in this picture, the injury during harvest created an entryway for mold. In this case, it's a Fusarium, and which can really collapse a melon within just a few days. There are many mold species involved, and even though they have a wide range of water activity, but individual species also have their preferred range or their limited range that they can simply cannot grow. Some common species here, Alternaria and Cladosporum are usually form those black or dark olive colored rot spot on vegetables. And if the bread mold usually are Mucro or Regipus, and Regipus also very aggressive, fast grower, can easily collapse like pumpkin within a day or two. And Fusarium is both plant pathogen and also spoilage agent. And Paecilomyces heat resistance. So sometimes you find that it can spoil lightly pasteurized food. And Penicillium aspergillus are two very common genera that just cause a lot of headache in the food industry. So let me briefly mention these two genera. Penicillium can grow in almost any environment with minimum available water and carbon source. Some of them may produce mycotoxin. They can tolerate low water activity. Some of them are xerophilic, and the temperature range is pretty high as well, from refrigerator to thermophilic condition, and the pH range is really high. So they do cause a lot of spoilage. If you notice, there's a green, like whether it's bagel, or whether it's bread, or even orange, if you see a bright green colonies, most likely Penicillium, occasionally Aspergillus, but Penicillium is more common in certain areas. Aspergillus is both humans' enemy and friend. The friend part is they have a lot of, they are used in biotechnology industry to produce enzymes and a lot of other functions. But the enemy part is they cause spoilage and also produce mycotoxin. They can easily degrade agricultural products before and after harvest. So for the crops after harvest, the water activity is low, but it's not low enough for Aspergillus to grow. And the mycotoxin they produce, including the really potent ones like aflatoxin and ochratoxin, and other not very potent ones. And they also have a wide range of temperature and pH that they can grow. Some Aspergillus species associated with ascospores that is thermotolerant, which is bad news for pasteurized food. And they typically can spoil anything, in the category of grains, dried beans, peanuts, tree nuts, and some spices. Fermentative yeast is an organism that makes ATP by aerobic respiration if oxygen is present, but is also capable of switching to fermentation when oxygen is absent. And the two examples are Saccharomyces cerevisiae. This is the one yeast. If you look at the range, the temperature range, the water active range, and especially pH range is pretty wide. And some strains even are preservative resistant, and they can spoil dairy product, juices, soft drinks, like salad dressing or Sprites, things like that. And Zygosaccharomyces Bailey and other species in this genus is very notoriously spoilage agent because their ability to grow in pH very low range, and also they produce gas when they grow, so they cause bulging, and they can easily spoil. If we contaminate it, it can easily spoil juice, honey, jams because of their ability to adapt to the food system. Oxidative yeast is a category that we call it film yeast, because when they grow, they're aerobic, so they tend to grow on the surface of things, of like a solution, or and then they form this film-like structure. Oxidation is a chemical reaction in which the electrons are lost and accompanied by reduction of gain of electrons. When we know that for acidified food, the pH is the control factor. However, some oxidative yeast and mold can spoil acidified food by metabolizing organic acid. If they grow substantially, they could raise pH of the food to an unsafe level. Lactic acid bacteria is a huge group. They're typically defined as gram-positive. They are rod or coccocyte. They are non-spore forming. Although they can be heat resistant, but they don't form spores. They are mesophilic, and in terms of oxygen, they're very diverse. They can grow in aerobic condition, they can grow in microaerobic philic condition, or they can be facultative anaerobes. So they can easily adapt, presence or absence of oxygen. There are two categories here. One is homofermentative lactic acid bacteria. Another is heterofermentative lactic acid bacteria. The difference is whether they produce carbon dioxide. If they do, they produce on the culture plate, they can produce bubble, and in the food, then they can make the food appear bulging. They do require high water activity to grow, but their pH range is on the acid side, is from 3.5 to 6.8 plus minus a couple of decimal points. And they may or may not show up on aerobic plate count. So in the lab, when we do testing, if a lactic bacteria is of concern, we have to use different media, not the typical aerobic plate count media. They are very common in the environment, and also very commonly found in food. They can spoil fresh meat, vegetable salad, fruit, liquid milk, or causing sour taste. And they also used in making product. For example, salami, sauerkraut, and cheese that people love, enjoy. Enterobacteriaceae is a very large family of gram-negative bacteria that has been studied and characterized for almost 100 years. And there are over 50 genera and over 200 species. And they are gram-negative and non-spore forming, and they can grow in temperature 18 to 47, but some of them maybe even lower. Their pH range is typically not acidified, not below 4.4. But of course, there's have plus minus a decimal point. But normally they do require water activity of 0.595 to grow. The examples of species in this family are E. coli, Citrobacter, Enterobacter, and Salmonella. So it's a very noticeable family and has a lot of species, and they can cause disease, and they can spoil fresh vegetables, meat, and poultry, fish, and eggs. Pseudomonas is is a genus of gram-negative rod, non-spore-forming bacteria. They are psychotrophic, meaning they can grow in low temperature. And they are aerobic and they're oxidase positive. They do require high water activity to grow. The pH range is 5.6 and above. So they are not typically considered acidified. And one of the things about Pseudomonas is that they can be restricted by sodium chloride. So if you make pickles, sauerkraut, one thing you want to do initially is add some salt to inhibit Pseudomonas growth, because species of Pseudomonas is actually pretty commonly found in vegetables. And if not controlled, they can spoil fresh vegetables, raw meat, poultry, seafood, and eggs. They usually cause discoloration and rot and really foul smell. Bacillus is a genus of gram-positive rod, aerobic or facultatively anaerobic spore formers. And they're mostly mesophilic. Some are psychotrophic or thermophilic. They require water activity of 0.9 to grow, and the pH range is about 4.5 or below 9.5. And because they produce spores, they are heat resistant. And some of the species here noted the Bacillus series, and if they grow in some strains of Bacillus series can produce intertoxin that can make a person sick. And another species, Bacillus subtilis, they can spoil bakery product, and but also some people use it as probiotic. Clostridium is if some genera or bacteria, they're good members than bad members, but it seems like Clostridium, all members, they are either bad or no good. And this is a typically gram-positive rod. They are obligate anaerobes. So oxygen will kill them. They don't need oxygen. They form spores. They can be mesophilic, psychotrophic, and thermophilic, meaning they do have a really wide range of temperature, can grow as a genus, right? But individual species might have narrower temperature range. They require water activity 0.9 to grow, and pH range typically we say is between 4.6 and 9.5. Although we have found some individual species can grow lower than that, and they are extremely heat resistant. The spores of Clostridium are extremely heat resistant, which is bad news for the canning industry. And when they do cause some species, when they do cause spoilage, they tend to smell really bad. But other species may not. So it's still a food safety concern. Lactobacillus gram-positive rod. They are microaerophilic, meaning they need a little bit of oxygen, but don't have to be the aerobic condition. They form spores, and they require high water activity growth, which means they usually grow in liquid. And they are commonly found in fruit and vegetable juice, and sometimes for even just a beverage that has no juice. If they use sugar, Lactobacillus come with a sugar, and they are extremely acidophile, meaning they can grow pH. They usually grow on pH lower than 6 point, or can as low as 2.0. They are moderate to obligate thermophilic. They grow under 45 degrees. So when you're looking at the ecological groups of microorganisms, you can tell what kind of food they can spoil. For example, the pasteurized milk that need to be kept in refrigerators, then you will find saccharotropes or spore formers that survived pasteurization that may spoil milk. And for cheese, normally cheese's water activity is pretty low. So you would find yeast mold or some lactic acid bacteria that may be able to spoil cheese. For fresh meats, can be easily attacked by Enterobacter AC, lactic acid bacteria, or saccharotropes. And then for fresh eggs is usually the whatever come with the contamination like Proteus, Seriata, Enterobacter, those can spoil the eggs. And for fresh vegetables, we see Enterobacter AC, Pseudomonas. Arvenia is a couple of species there can spoil fresh vegetable, make it really smell bad. And mold, of course, spoil low water activity and low water activity that is beyond what the bacteria can grow, then mold can attack. So how microorganisms cause food spoilage? What we call food spoilage is really when microorganisms eating our food. From the microorganism point of view, I mean, if they do have one, there's nothing wrong with it. As a matter of fact, their ability to break down organic matters plays a critically important role in nutrient recycling in nature. To digest different types of nutrients, microorganisms evolved ability to produce various enzymes and metabolic pathways. These biochemical reactions are the mechanism that changes the characteristic of food. For example, sugar and carbohydrates are digested by cellular respiration or fermentation. Protein can be digested by proteolysis process, and fat can be digested by lipolysis process. And of course, microbial growth on food surface and the toxin production are also ways for causing spoilage. Most microorganisms oxidize carbohydrate as their primary source of cellular energy, basically carbon. Cellular respiration is the process to release energy by breaking down glucose and other food molecules in the presence of oxygen. Where fermentation is the process by which cells release energy in the absence of oxygen. And then there's lactic acid fermentation, which is a metabolic process by which glucose or other like 6 carbon sugars or 2 carbon sugars are converted into cellular energy and lactic acid. And we mentioned heterofermentative and homofermentative. Fermentation produces variety of products, including ethanol, acetic acid, lactic acid, and carbon dioxide. Is basically a mechanism that support the entire industry of anything that relating to fermentation. This fermentation process produces many desirable products, such as soy sauce, cheese, yogurt, wine, and bread. When fermentation is not desired, then it's a spoilage, right? For example, you may enjoy yogurt, but you would not like to drink soured milk. Pectic substance are important cell wall components of higher plants, basically, like vegetables and fruits. They kind of the glue that connect the cell wall together. Bacteria and fungi can break down this glue as well, so that will cause the collapse of the cell wall and cause the rotten of the vegetable. To break down protein, though, you need proteolytic enzymes and bacteria that has that. Proteolysis is the breakdown of protein or peptide into amino acids. by the action of enzymes. Spoilage bacteria produce protein-lytic enzymes, and they break down, foods such as meat, milk, or seafood. The compound produced by proteolysis and related metabolic activity often has a really bad smell. That's because the chemicals they produced during this process. This process is highly regulated and fast in the presence of water. So that's why, like a fresh meat has a lot of water inside, they can go really bad quickly. And some species, like if it's Pseudomonas, then it produces even more bad smell chemicals that just keep people, animal away. And for fat, bacteria produce lipolytic enzyme to attack it. Lipolysis is the breakdown of fat and other lipids by lipolytic enzymes, and the end product is fatty acid. And many microorganisms produce these enzymes, such as Pseudomonas, Staphylococcus, Rhizopus, Microbial, and the fatty acid they produce in the end of metabolic pathway usually give this off flavor or really rinsing odor. Most of the microorganisms can spoil food by growth on the surface. They simply grow and accumulate biomass on food and making the food unsuitable for consumption. So that's including mold, yeast, and bacteria. Some of those produce mycotoxin, and the mycotoxins are toxic compounds that naturally produced by certain types of mold. Mold that can produce mycotoxin grow on many different types of foodstuffs, such as cereal, dried foods, nuts, and spice. Mold growth can occur either before or after harvest, during storage, in or on the food itself, and usually under warm, damp, and humid condition. And most of mycotoxins are chemically stable and can survive in food process. Bacteria can also produce a toxin. One of them, CBOT, those toxins are really lethal. And also they're intratoxin and neurotoxin. So in summary, we have discussed that microorganisms can break down food in different ways, including proteolysis, which is breakdown of protein, and cellular respiration, which is breakdown of sugar in the presence of oxygen, and fermentation, which is breakdown of sugar without oxygen, and lipolysis, which is breakdown of lipid, and then of course surface growth. Often bacteria and fungi can attack food using one or more mechanisms. That's depending on the type of the food or the food processing method. Note that the picture shows with fresh milk, meat, or fish. If the food are processed, then the spoilage mechanism will change. For example, bacteria can easily attack fresh meat, causing undesirable sensory changes, right? Cooked meat, where on the surface, the bacteria were eliminated or killed or reduced, but cooked meat can go bad due to lactic acid bacteria. And if the food is made into jerky that has, really, really low water activity, that can really inhibit the microbial growth. But if the jerky is not properly made, and if there's just still certain water activity that's sufficient for mold growth, especially osmophilic mold, then they might find a way to spoil jerky. So when you evaluate shelf life of food, it is important to understand that what to begin with. So what do you have there? What goes into the food and what is going on inside the food during the storage? And then assess what to expect in terms of stability of the food. For example, ingredients might carry microbial load with them and then go into the food. The process may reduce or sometimes may even increase the microbial load. And during the production time, the machinery, the people, the environment might introduce microorganisms into the food as well. After production, the intrinsic factors such as pH, water activity, salt, fat content of food, and extrinsic factors such as packaging and storage temperature might impact the stability of the food. Let's say that a raw and processed food become contaminated by microorganisms, but the food itself has defense system, such as high acidity, low water activity, etc. Only a part of its primary microflora will survive under the selective pressure. The microorganism that can adapt to the food without the kind of tough food will grow. A particular microbial community will develop and which will cause the spoilage. So for example, if we take the orange, the fruit orange itself, and the orange skin is really to protect the orange, right? And the orange itself has, the skin itself has essential oils and the orange itself is on the acid side. So mostly the fruit, when it's intact, you can preserve it for a while. But when the immune, after harvest for a long time, the immune system is low, or if there is injury to the skin of the orange, then mold can attack it. But you would think, mold is mold, right? They can attack any type of food. But actually the orange itself, because of low pH, is have selective pressure. So only some species of mold can associate with spoilage of orange. So in this case, we often found Penicillium digitatum or Penicillium inoculum appeared orange, not everything. But if the orange is so far gone, like almost gone, like the Penicillium attacked first, and then the condition is improved for the other mold, you might find other mold. But initially, Penicillium is the cause of spoilage. So let's talk about the factors that really control the microbial spoilage, which is the intrinsic factor of the food itself. And then we'll talk about extrinsic factor. Water activity of food is the ratio between the vapor pressure of the food itself and the vapor pressure of distilled water under identical conditions. You can think of water activity as an indicator of how much free water molecules that bacteria or fungi can use for their metabolism. If the water molecules are bounded by salt or sugar, then the microorganism will not be able to grow. As we discussed earlier, that bacteria and fungi have pH requirement. So controlling pH of the food, you can control microbial growth. You can also use presence of and absence of oxygen to control microbial growth. For example, anaerobic condition will prevent obligate aerobic bacteria to grow. So the aerobic environment will not be suitable for obligate anaerobics. In food industry, various technologies are used to prevent microbial growth and to prevent spoilage to extend shelf life, such as thermal process that reduce the microbial load or eliminate them altogether. Non-thermal process such as filtration or HPP, and there's a filtration or refrigeration or freezing to slow down the microbial growth. And then of course there's vacuum and modified air packaging to prevent spoilage due to aerobic respiration. Food microbiologist knows about hurdle technology that helps obtain microbial safety and the stability of the product, and at the same time maintains organoleptic properties of the food. Hurdle technology is a method of ensuring that pathogens in food products can be eliminated or controlled by combining more than one approach. For example, you can use pH or water activity or temperature, or salt level, or combinations of those to prevent microbial spoilage. You can also combine intrinsic and extrinsic factor. For example, keeping kimchi in the refrigerated condition, right? It's a low pH, but you want to keep it in the refrigerated condition. This is a summary of principal hurdles used for food preservation. Like drying, curing, or preserving, or effort to reduce the water activity. Acidification of formulation is effort to control the pH. And then heating, freezing, refrigeration is using the principle of temperature range, and packaging like vacuum, modified air packaging is a way of alter the oxygen availability. And then of course there's a chemical preservatives that people can use. So if you're looking at this picture, there were several different type of foods. What are the control factors for this food, right? For canned food, for yogurt and dairy products, for yogurt and dairy products, what do we know? We know they do pasteurize the milk, and then they inoculated milk with probiotics that can fermentation, lactic acid bacteria fermentation. They also have other species to add in the flavor to optimize the process. And also yogurts meant to be kept in the refrigeration. So it's multiple hurdles used in this case. For baked goods, so the initial process, the baking will reduce the microbial load, right? And then the baked good usually very low water activity to keep it dry to prevent mold from growing. And then of course, acidified foods use pH to control growth. And same thing with probiotic drinks. Probiotic drinks usually have low pH, and also they might have live cultures that keep pH low. Then let's look at some examples in more detail, like salad bowl. So when you have this salad bowl, what are the ways they try to extend shelf life? For example, in this the picture, the chicken is cooked, the cheese are processed, low water activity, the corns are grilled, cooked, and then salad dressing is most likely acidified dressing, and the vegetables is washed. So in this case, which components or ingredients determines shelf life? The ones that goes bad first, right? If you purchase salad bowl for lunch, if it go bad, you throw away. But which of this component can go bad, right? In my experience, is usually the vegetable, because the washing itself can reduce the microbial load but not eliminate it. And so, what is the control? How we can extend shelf life of salad bowl? Which is normally we see is the refrigeration. You can keep it in the refrigerator for 5-7 days. In beverage, the processing of beverage, including microbial reduction steps, right? Like water treatment, filtration, and some beverages have pasteurization process. And then the controlling factor usually is pH. They keep the pH very low. And then in some beverages, they add preservatives. And then for soft drinks, they use carbonation to inhibit microbial growth. And then in some other beverages, like fruit juice based, they may require refrigeration. how about butter? What's the spoilage mechanism of butter? So butter usually have low water activity and high because of high fat content. So they're not easy, they cannot be easily contaminated or spoiled by microorganism. However, if you add things to the butter, if you add fresh herbs, that's actually increase the baseline microbial load. And what happened to those bacteria or fungi introduced by the fresh herbs? That's something we need to look at. Would they change the water activity of that niche area so they can have a really small space for them to grow, or the low water activity of the butter really help it prevented them from growth. And also you can add salt to the butter to as a measure to inhibit the reduce microbial growth potential. And some butter, most of butter, we still say keep it refrigerated. Salad dressing, the control factors of salad dressing usually low pH. Low pH is good control most of bacteria, but we know yeast, mold, and some lactic acid bacteria can tolerate low pH, so they may be able to grow. So in some salad, they use preservatives to help prevent spoilage. And then they can also refine the process of making the salad dressing, like hot fill or other water treatment, other things, the environmental control. And then some salad dressing may require refrigeration to ensure the shelf life. We usually say canned food is safe because the two factors there is thermal processed to ensure the kill of, if not all, then most of the bacteria. They may some spore formers survive, but mostly we say the thermal process is through, is validated, and then the hermetic seal, right? They should not allow any bacteria or fungi enter the can after products released to the market. However, if something goes wrong during the process, then you will have spoilage on your hand. For example, if the thermal processing is insufficient, then some spores survived and they were able to grow. If the cooling is insufficient, then thermophilic spore formers can germinate and grow. And if there's a post-processing contamination, for example, the can has seals not properly, has liquor spoilage, and also if you receive the ingredients that already spoiled, and then you have intertoxin there, and the process cannot kill intertoxin, you have your not only spoilage but the food safety concern. So that summarizes my talk today. We have about 10 minutes left. I'll be happy to answer any questions you may have. I'll do my best, try to answer. If I don't know the answer right away, we'll have, send us email and I can look up literature to give you a good answer.
Thank you so much, Florence. We do have a few questions that came in. If you guys want, you can keep putting questions in the chat or the Q&A button. The first question is, for Enterobacteriaceae, what would be the target limits of in-spec counts for a sponge swab? What would be considered an out-of-spec limit range?
For the environmental sampling? Or
Yeah,
we talk about
Enterobacteriaceae.
So, as a microbiologist, we usually shy away of giving you exact number. But if I hate to say depends, but there are different levels we talk about. So we talk about environmental sponges, right? And after sanitation and of a surface, if it's a food contact surface or if it's a zone 2, environmental samples after sanitation, I would say you shoot for, if it's a clean processing plant, I would say you shoot for less than 10. But what less than 10 what? A lot of standard didn't say, when you talk about number, you got to have a unit. So less than 10, maybe 100 square centimeters, right? So have that, or if you say, sometimes there's incidental contamination during sampling or handling. So maybe less than 50 as a goal, but give it a range. If the facility is fresh produce, the number interbacterial acid there in the fresh produce can be pretty high. Then less than 10 or less than 50, maybe a goal that's not easily achievable, then you can have a higher range. So it's really based on your understanding, your baseline data, right? You don't, I'm hesitating to give a hard number without knowing the actual facility, but I'm happy to help. If you send me some baseline data, I'm help and tell me what's the process, whether it's, what's the sanitation process, I'll be happy to work that out for you or with you.
Sorry, I was on mute. The next question is, what microorganisms can spoil plant-based meats?
So the plant-based meat, you see the, one of the slides we talk about, when you think about that, you always think about what comes in, what is going on, and then you can access what would happen. For example, if you use soybean as the main component, then what is the microbial load in soybean? And then during your process, if you don't have a cook step, if you just grinding, mixing, then everything, the bacteria come with the soybean will be the spoilage agent if they keep the meat in the room temperature, right? The soybean, when it's dry, it might come with a microbial load, but when they dry, the microbial just there, they don't grow. But once you're mixing it in your process of making the meat, then suddenly you add water, you add, some material look like blood or something, the water activity is high. So they will spoil the food. And other ingredients, whether seasoning, flavoring, whatever come into it, they carry the microbial with them. And if you do nothing, if you're just mixing them, and all those microorganisms can potentially spoil the plant-based meat. That's when we see, some of our clients making plant-based meat, they keep it frozen, right? Just as if you would handle animal meat. They will spoil like animal meat, and in some cases they may even smell as bad as animal meat when they spoil. Cellular, cell-based meat is different story, right? This for animal cell to grow in the condition, they have to keep the condition sterile, meaning they cannot have bacteria and fungi during production. So the microbial load there is low. But then the contamination would come from the environment, right? The aseptic production is very hard to achieve, very, very hard. So anything coming to the food in contact will be the agent to spoil cell-based meat. That's why cell-based meat still need to be kept in the refrigerator or freezer.
Thank you, Florence. We've got a number of questions here, so I will try to get them all answered. But if not, you can reach out to us or we can reach out to you and get them answered that way. The next question is, what are examples of potential water treatment for dressings to improve shelf life? UV.
So we talk about water treatment.
Yeah, what are potential water treatments for dressings? For dressing products to improve
You know,
shelf
if it's a pure water, the best treatment is membrane filtration, is filtration. So the filtration can remove the bacteria, right? I'm not sure about how effective UV in terms of eliminate bacteria. It depends on the volume of the tank, or whether UV can penetrate throughout the water tank, you know. But the best way I see for water treatment is actually filtration.
All right, next one. What would be the best to aid in the control of Clostridium?
Best to control Clostridium?
Yeah.
Normally people use like pH is one control, right? For example, we know Clostridium, you know, they're heat resistant, so heating might not completely kill them off, but they require, they're mostly mesophilic. There are some species that are not mesophilic, but learn the Clostridium is pretty diverse genus. So there are some species mesophilic, some species can grow in psychotrophic condition, and that most of them require pH above a certain level, like 4.6. So you can use pH to control it. You can use, they cannot grow when there's oxygen, so you can use aerobic condition, and then you can also use temperature. Depends on the species.
Okay. The next question, sorry, just trying to go through these quickly so we can get to a bunch. Is it possible, by the way, yes, the presentation will be shared with the participants, or the video will be. Is it possible to have spoilage from bacterial and fungal sources at the same time?
Yes. That's kind of depends on the food, right? For example, if we talk about finished good, then you can have bacteria grow there, like the S bacteria. You can have yeast, and if you test all three, like Arabic plague count, like the S bacteria count, yeast mold, they all high, so they can grow. In other cases, for example, like in meat, really fresh, high water activity, protein rich, fat, you know, like mainly protein rich, like meat, then bacteria will grow so fast that they, like yeast and mold, didn't get a chance, right? Even though they can grow, but bacteria will occupy the surface, and they're the first colonizer. And then in other foods, you know, when the water activity low, then yeast and mold can attack it, and bacteria didn't have, you know, don't have a chance. So it really depends on, again, depends on the food and the conditions it provides, whether it's, you know, in terms of low pH and water activity and all that. If you're giving the chance, you know, if the condition is good, you know, microbial organism will grow. You know, that's what they do.
Okay, next one, any recommendations on control factors on enzymes?
On enzymes?
Enzymes. Any recommendations for control factors on enzymes?
The enzyme from the food or enzyme from the microorganism?
That's a good question.
We'll
follow up with you.
Yeah, because I need to get more information about that question, because the control enzyme produced by microorganisms, you control the microorganism. You know, it's hard to, you know, it's another science to alter metabolism of microorganisms, you probably want to go easier way, which is just control the microorganism. But for food enzyme, that's not my expertise.
Okay, next, we'll do one more question, and then we'll wrap up. How is it that making dairy products requires lactobacilli, but at the same time, these bacteria can cause spoilage? What is the determinant factor?
Can you repeat that question again? So the
Yeah, basically, it's the
Yeah.
lactobacilli is required for making dairy products, but it's also a bacteria that causes spoilage. So what determines whether it's used for making the product versus
you want to, right? So it's about control. So if you know the lactic acid bacteria come with the milk, especially the milk is easy. Their type of milk that don't spoil. Actually, they have some treatment. You don't know what the species of lactic bacteria, and you don't know what what the flavors, what the products they would generate. The yogurt production is a very sophisticated industry. They control the strains of the lactic bacteria they use. They also add the different species to improve the texture. You see the yogurt had that perfect texture. They can control the flavor profile. So it's all about controlling, right? But if you're making pickles, then you basically tap into the naturally occurred lactic acid bacteria to grow. And then there's certain ways to making pickles. First, you want to reduce the microbial load by either blanching the vegetable and add salt, and then you remove the air, so it inhibits aerobic bacteria from growing, and that's promoted growth. So it's about controlling.
Thank you. We only have one more question, so I'm gonna, if anyone needs to leave, feel free. But we'll do this last one. Sometimes bakery items remain looking good for a long time. Is it really possible that they have not spoiled for months?
They probably add the preservatives, and they're probably very dry. So you keep it dry, so even mold will not be able to grow. Or you add preservatives if it's not that dry. So there are ways to control it. You probably see like cookies. If they're so dry, you won't see microbial spoilage. But over time, you might have rancid because there's a fat inside. The cookie may taste like stale and rancid. So chemical spoilage is, of course, another factor that causes the loss in food.
Thank you so much, Florence. With that, we are out of time for today. I want to thank everyone again for joining us. If we didn't get to your question, or if you have one still, please feel free to shoot an email to lab@amtec.com. We'll be sure to get back to you with an answer. Again, thank you so much for attending, and we look forward to seeing you at part 2 of our shelf life series in August on the topic of study design. Thank you.
Yeah, thank you, everybody.